So we're going to jump right in, but it's a kind of broad spectrum. Mine will be a lot broader in the sense I'll talk about our omics and deep data profiling in general, maybe set the stage in a broader context for, at least the field or what we're doing, big data. And then it's quite a spectrum of different proteomics technologies and talks we're going to have. So I think that's going to make it a lot of fun, and hopefully we can learn some of the latest and greatest things out there. So I am going to talk about our work as say using deep data to try and transform healthcare. So some of you may have heard me talk about this before, so I did add a few new slides. I know for those of you going to the HUPO meeting, I'm going to try and give a completely different talk when we get there, based on some other new data that's hopefully coming in. All right, and some analyses being done. So anyway, many of you have heard me say this before, but our healthcare system, we believe, is broken. We really practice sick care, not healthcare. And even when we practice healthcare, we do it in a very, very antiquated fashion, in my mind. The idea of traveling to a doctor's office for routine tests, I think we'll still need that, but I think the degree should diminish rather rapidly. And then the idea of going in, they take lots of all this blood and other measurements, and then or blood and other measurements. But at the end of it, they don't make that many measurements from all the time you're there and the information and the samples they collect. And then they treat you based on population averages. And I think we really want to change all of that. And that is our plan. So I like to put this out there because I do think it sets a stage. Your health is impacted by many, many different things. It's impacted by your genome. That really only accounts for about 16% of your lifespan, although I just came from a meeting. If you're one of these centenarians, it counts for more of your lifespan, meaning it's more like 60%. Okay, so what I was saying is that we're trying to fix the broken healthcare system, all these steps that we think are pretty antiquated, when you could actually be doing a lot, and we'll talk about this monitoring at home. And I think the omics technologies will be particularly powerful for, certainly from the research side, but I think also for the clinical side as well. So I was saying that your health is influenced by many things. Your genome counts for only about 16% for most people, but for those who wind up as centenarians, it is a bigger factor, but still there's a large component from other sources as well. And in particular, your lifestyle, like activity, food, mental health, as well as environmental exposures, we know all impact your health. And we're in a world where we can now quantify a lot of this. And so even if we don't quantify it directly, we can try and quantify its effects by doing deep data dives on people. And certainly for lifestyle, that's actually easy to measure. And so as many of you know, we set up this personal omics profiling project many years ago. And the idea is to collect very, very deep data on people. We'll sequence their genome once. And then from blood, urine, and then stool, we'll actually measure many other things. In particular, from blood, we'll measure out of peripheral blood monocyte cells, we'll measure transcriptome, proteome. And on some individuals, we're trying to scale this more for the epigenome. We're also measuring out of plasma the proteome, cytokines, metabolites, and lipids. And we'll measure these out of urine. Microbiome, we've just measured out of 4 areas. The gut is most popular, but nasal, skin, and tongue, we just had a paper out on that. That's pretty interesting, where they're all very somewhat co-associated and co-coordinated and probably very tied into your immune system and other health aspects. So we do this on top of the clinical tests, questionnaires, and various advanced tests, stress echocardiograms, and glucose control measurements. So I'll talk about this a little bit in more detail. And then wearables, we're very, very keen on these because they're continuous monitors and very powerful for tracking health. So one aspect is to do very, very deep data dives on people. And then the other aspect is to track them longitudinally. So we sample people every 3 months while they're healthy, and then take more samples when they get viral infections and other things happening. And this started out with a cohort of 109 people. Some have now dropped off, but we're somewhere on the order of about 200 people. And we're doing this partly to understand what a healthy profile looks like. How does it change over time? And what's the difference between people? And we'll have a project we'll be putting that we're trying to finish up to look at not only differences between people, these are all from the Bay Area, but also from around the world, a project we've called HPOP that the US, actually, sorry, the Human Proteome Organization was part of, and we think that's kind of an exciting project, community project. And so we're trying to understand that diversity, but we're also trying to understand if we can use these advanced technologies like genome sequencing, wearables, and proteomics to some extent, can these be used for better managing people's health? And with regard to that point, basically the answer is yes. By doing very, very deep data dives on people, we can make many health discoveries, and these are just from the first 3.5 years. We found a lot of very important things spanning many different areas: hematology, cardiovascular, metabolic. And all these conditions were found pre-symptomatically. So nearly half the people learned something pretty important about their health. Again, all pre-symptomatically, and some of them were a big deal, like early lymphoma. These two are pre-cancers that can convert to aggressive cancers. We found 2 people with serious heart issues, one from genome sequencing, one from wearables. And that was another lesson. No one technology found all these things. By doing the whole host of technologies, we got a much, much better picture of people's health. And as a consequence, we're able to catch these things all pre-symptomatically. And so the analogy we like to use is that if your health is a 1,000-piece jigsaw puzzle, we're trying to get 600 or 700 pieces versus we would say 5 or 6 we measure today. We just don't do very, very deep data measurements on people. So one area that was important was the genome sequencing. 12 of the first 70 individuals had an important mutation. These are what are called single Mendelian mutations that put people at high risk. And the prototype is BRCA. Women with a BRCA mutation are high risk for breast or ovarian cancer. Men are at high risk for cancer too. And there were other things found as well. And some of these turned out to be, again, a big deal. This individual is a young fellow and actually has a heart defect that was found from the genome sequence. And another person had a mutation here, and they actually had early thyroid cancer. They did a whole body MRI. Now, more recently, this is single gene mutations. Most complex diseases are not single gene mutations. They're actually they're thought to be due to common variants of small effect. And people have done these GWAS studies, genome-wide association studies. You may be familiar. I know this proteomics crowd. But with GWAS, you can actually make these associations. And the problem with GWAS is it doesn't work very well. You can't get good predictive models, maybe the top 2 to 4%. And we think that's in part due to the fact that they ignore rare variants. And they also don't, they assume genetics is linear. And for anyone who's done genetics, it's not linear. 1 + 1 is almost never 2. Some effects are synergistic and much higher, and others are lower. So I won't have time to get into this in detail, but we came up with new machine learning methods. You can actually look at the references here. And with these methods, some are based on rare variants, and these are based on GWAS studies. We can find usually 10 to 100 times more genes with these methods and validate them by various means. And I think here's where actually protein networks were very useful for the validation. And as I say, I won't have time to get into this particular one, but these new methods we think are going to be super powerful. This was carried up by Saizong and Jingjing Li when they're in the lab. And then more recently, we've set up a version of this that I want to give you a flavor for this, where it's the same idea, but we map out regulatory regions, which is where most GWAS signals are thought to lie. And then we basically can take single cell data for each of these open chromatin regions, these regulatory regions, and using machine learning and GWAS signals, you can actually integrate all this information and say what individual cells have what signals for genetic, if you will, variants that's associated with the disease. And this turns out to work really, really well. It's much better than traditional methods. Again, there won't be time to go through this in detail, but it's an it's a deep learning method, this one is, for trying to actually integrate, again, single cell and GWAS information. And the net result is that you can come up and make some predictive models about disease risk from these new scores we're generating. You can also, because you're dealing with single cell information, see what cells carry most of the signal for the genetic. Now, this is the genetic basis of these diseases. And then you can get in there and actually get in, what genes are involved, what pathways, and that sort of thing. And so we've just applied this to several Alzheimer's, cardiomyopathy, and then I'll show you one example with type 2 diabetes, pretty well trodden area with lots and lots of GWAS studies out there. We took a GWAS study that someone had generated and ran this approach. And the bottom line is there were two cell types that have most of the genetic signal. One is the beta cells, which, excuse me, we expected. We can even go into the subtype of beta cells, but also the alpha cells turn out to have a lot of the type 2 diabetes signal. So we can now use genetics to define, what cell types are really having an important role in the genetic basis of this disease. And then we can go on and see, what genes are there. And it turns out half the genes you find with this new AI method, they're new. They actually weren't found, even though there were these big, literally hundreds of thousands of people, cases and control studies. We can still find half the genes we find are new. And as I say, it points smoking guns at cell types that had been implicated, but I would argue had not been that well pinned down. So we think there's new areas for genetics. Now, one reason I brought this up is that this is just part of the equation. I think if we can integrate proteomics and metabolomics information on top of this, we're not there yet with predictive models. And that's where I think if we bring these other data types, we will get there. That's my optimism. Okay. The other thing we've been doing by profiling these people over time is that we can see how they're changing, which we interpret as aging, especially when we look at just the healthy time points. And what we discovered is that everybody ages differently. So this person, their top biochemical pathway that's changing is their cardiac hypertrophic signaling pathway. For this person, it's their immune and metabolic pathway. And we wound up classifying the people into what we call ageotypes, their aging patterns. And so there are four major patterns: kidney, liver, metabolic, and immune, as defined at this time. We didn't have so many people. We have more now. We know there are more ageotypes than this. And so each column is a different person. Each row is an ageotype. And you'll notice that some people age in all four categories. This person ages in three of the four, and so on. So we can see exactly how people are aging. And then I'm going to come back to this at the end, but there's actionable information around this, meaning they're clinical markers. And some people have actually acted upon this, exercise whatever, for example, and changed their metabolic ageotype. Now, I'm not saying they got younger, but I do think they've improved their health. Okay. And then the other thing we've done more recently, and this seems to have gotten a lot of attention, is that we've taken the group as a whole and we say, well, are there certain periods where there seem to be more changes than others? And it turns out, so by actually looking across these different age categories, there are changes across all the different age groups, of course, but we notice that there's a big burst in the 44 is this one, and this one's around 60. So now 44 is a little precise; it's a little broader than that. But you get the idea that we think aging is not linear. That, and in fact, if you look at the pathways, will change at all different times throughout this. But there are two particularly impressive bursts, shall we say, one in the 40s and one in the 60s. Now, most audiences you're talking to, they're either on their way to the 40s, in their 40s, but they're out of their 40s on their way to the 60s. So everybody sort of wonders, well, how's this relevant to me? Well, here's how. We can see your biochemical pathways that are changing. And it turns out, by the way, I should say that the changes in the 60s we did expect, right? Disease associations go up and your immune system declines. Your risk for infectious disease. And we do see a decline in immune pathways, and particular viral pathways are shifting here. And we did see changes in skin and muscle. Again, things you would expect. We also saw changes in kidney function and carbohydrate metabolism. I think this occurs throughout, but particularly acute as people hit their 60s. The 40s, in hindsight, you could expect it. I'm not sure we knew offhand that would be the case. But same thing, we did see changes in skin and muscle mass. So a lot of these are extracellular matrix proteins, it turns out. and some key muscle proteins we're picking up. Cardiovascular disease is about alcohol and caffeine metabolism, and then lipid metabolism. And I think that in the end, a lot of these things do make sense. People's LDL does seem to increase then, and there are changes in people's fat deposition as they hit their 40s. So again, I think intuitively it makes sense, but the nice thing is we're kind of pulling the curtain back and seeing in a lot more detail what pathways are shifting. I still think it's most important to know what's going on at the personal level, but we think this is a global thing for physicians and others to be on the alert for. And maybe when you start seeing your statin, your LDL shift, you start going on Lipitor for these statins a little bit earlier than you might otherwise do. Now, one question, this is a research project, as you've noticed, and one question I get asked a lot is how are you going to scale this? And so, we're in Silicon Valley, so the way you scale these things is form a company. And I think, some of you have heard me say it before, I think academics were really good at discovery and proof of principle. We're not that good at scaling. Many think they are, but they're not. Companies can do this stuff just way, way better. And so we formed a company that does a medical version of this. And it's much more actionable the way it's set up. Right? Again, what I showed before was a discovery project. And so they do a medical version, what I said, but they also do whole body MRI. And just from the first 100 plus people, they found pretty important things. I should say both cohorts are in their 50s, most people, although it's a spread. And this group, they found some of the early ovarian cancer, prostate, even early pancreatic cancer, which is picked up by longitudinal profiling. And then cardiovascular things and such as well. So, and these are all found, once again, pre-symptomatically. So the idea that you can do these deep data dives and see things in incredible detail, we think is powerful. And what I like about this is that I think we can get this inexpensive, meaning, and this is pricey now, it's $3,500, but I'm quite confident we can get this much, much cheaper going forward once we, keep improving the technologies and get the investors back their money, that kind of thing. So this is where we're hoping to take this. All right. So the other area we're pushing very, very hard on is this idea about Amazonizing healthcare, trying to do remote monitoring. And I think and this helps open up the omics field quite a bit. If the idea is you can collect samples remotely and then analyze them offline in other locations, it'll be very, very powerful. And in fact, so we think we're hoping to use this to both transform healthcare, but also we're running a lot of our research studies this way now. It's a lot cheaper. You're actually taking samples and measuring people at home in a more native environment. And I think as such, the measurement should be a lot more reliable. And so the two approaches we're using are wearables and this home microsampling. And so for the wearables, you may know there's over 1,000 devices out there. Many are in the form of smartwatches or rings. And, I wear a lot of these things. I'm down a watch. There's one broke, and I also lost my hearing aids. Hearing aids, I think, are going to be very, very powerful as wearables in the future, especially given we have an older population. And they don't just measure. I wear mine for hearing, by the way. So if I have trouble with the Q&A, it's because of that. But I use them for monitoring too. And what's powerful about all these devices is they give passive monitoring, meaning they're following you all the time. And some of the measurements are quite accurate, heart rate variability. And some of the other measurements, they may not be accurate, but the deltas are fine, like blood oxygen and what are some of the other things? Blood oxygen, something called conductance, galvanic stress response, which is skin conductance. It turns out to be a pretty useful measurement, and they never measure that in a physician's office. And so there's quite a few measurements you can get from these devices. Again, some we think quite unique and powerful. And some of you may know this, but we discovered early on, actually, that you could detect when someone gets an infectious disease. In this case, it was when I got my Lyme disease in 2015, and we picked that up because my heart rate went up and my blood oxygen dropped. And that was pretty easy to spot with a pulse ox, as shown here, and a smartwatch. I later learned my skin temperature was off as well. And so we were able to see that. And then we went on to show that you could tell respiratory viral infections actually from a simple smartwatch as well. because your heart rate will jump up. And these are all in advance of symptoms, okay? So you actually see this jump up in physiology before symptoms. And then we did publish that a while ago, 2017. And of course, along came the COVID pandemic. I won't spend a lot of time on this, but although I would still like to have all of you, oops, somewhere it says, yes, so one of these others, love to have you all sign up in our studies, by the way. How many of you are wearing a smartwatch or ring or something like that? Oh yeah, good crowd, excellent. So do sign up in our studies. So we went on to basically show we could detect COVID with this, a median of 3 days in advance of symptoms from a real-time alerting. And that's in fact what we built. We built a real-time alerting system that tracks your basically your profile in a circadian fashion. If you have a jump up in signal for too high for too long, it sends off a red alert. You actually have to click it soon. I hope it'll come back to you automatically. And it works. So it works a median of 3 days prior to symptom onset. This is one of our first cases. Person got a red alert here. They were getting red alerts for 3 weeks prior to that. Sorry, this is when they got symptoms. They were diagnosed the next day. It works 80% of the time. We're not there 100%, and that's one reason we want to pull in more data, get more people. I know we can improve this. This is mostly built around resting heart rate. And it's very, very sensitive. It picks up asymptomatic cases as well. We think you need as little as 2 beats per minute increase, and we'll be able to tell. Now, it's not specific for COVID. It's really more of a general stressor alert. And mental health stress, workplace stress is the #1 trigger of red alerts. So, yeah, basically your heart rate does go up. And this is why we want to also have more people joining, because I'm quite confident we can tell the difference between a respiratory viral infection and workplace stress, because, you know, if we brought a respiration rate and things like that, they'll get very, very different signals. Some of these other things will trigger red alerts too. Most of these you can contextualize. Like if you run a marathon, your heart rate will be up for several days, and that will trigger red alerts. But I think this is, again, the tip of the iceberg, because if you think about these devices, they're shining light into your blood. They're measuring blood, and they're measuring, for example, oxygenated hemoglobin is measured by a spectroscopic shift. And so they can make a lot of measurements right off your blood. And with machine learning, we can ask what features on a watch can be used to predict clinical measurements. This is red blood cell count, hemoglobin. These are relevant for anemia. This is A1C and fasting glucose, relevant for diabetes. So we can actually get decent signals from a smartwatch. This is a 2016 smartwatch. So I'm quite confident we can do better now with the higher resolution devices. And so I think these are going to be super powerful. The other area we do a lot with is glucose monitoring. I don't know how many of you have worn a glucose monitor. Yeah, a few. They're now over the counter, actually. You may know that. And so they're very, very powerful devices. They measure your glucose every 5 minutes. And we got involved with this when they're mostly just being used for insulin-dependent type 1 and type 2 diabetics. We started putting them on so-called normal and pre-diabetics. And we discovered a lot of normal people are normal. Some, though, are moderate spikers, and some are severe spikers in their glucose, probably just as bad as diabetics, actually. And then we classified people, we came up with the algorithms, classified people into glucotypes, reflective of their glucose levels. And then we, as did Aaron Siegel, went on to show that different people spike to different foods. So some people spike to bananas, some to pasta, some to white bread and brown bread, what have you. And this is from Aaron's work. He showed here's a person who spikes a banana but not a cookie. This person spikes a cookie but not a banana. We show people spike to bread and peanut butter, others protein bars, which, by the way, can have lots of carbs in them. Don't let them fool you. And then cornflakes and milk. You know, nearly everybody spikes with cornflakes and milk. That's so super evil. Watch out for it. Okay. Here's one of our latest experiments, which I think is a lot of fun. This is done by UA in the lab. So he took 55 people. They're wearing CGMs, and we gave them the exact same amount of total carbs, but in 7 different forms. So mixed berries, grapes, rice, and so on. And found the same thing, that again, in a more well-controlled experiment, different people spike to different foods. So this person, blue, is bread. He's a bread spiker. This person's a grape spiker. This is pasta. This is potatoes and rice. Most people, by the way, are in this category. They're rice spikers, just to warn you. White rice is also pretty severely glycemic to most people. And then we wanted to understand this in more detail. And if you think about it, diabetes is very, very heterogeneous, and it's an area that I think is ripe for picking with omics technologies, including proteomics. And so it's heterogeneous probably because it's a very complex system, right? It involves your liver, involves your pancreas, your muscle, even your brain is a major glucose metabolism organ. So all these different tissues, all these different biochemical pathways are involved, and some of them becoming very famous. Incretin is a pretty important molecule. It's what Ozempic is an analog of, if you will. And so you can actually now subclassify diabetes based on the defects like insulin resistance or beta cell defect or other parameters. And actually there's several forms of insulin resistance. There's muscle, there's liver, there's fat. And you can also subclassify it based on people's response to drugs and also their biochemical defects. People are now looking at molecular profiles. And so we wanted to see if we could relate subtypes to food and other parameters. I won't get into this detail because it gets more metabolomics and proteomics, but we did subtype people with very gold standard tests for muscle resistance, beta cell defects, incretin, whether they're having an incretin defect or not, hepatic, and so on. And people do have different forms of diabetes. Again, this is a heat map. Each row is a different person. This person has an incretin defect. These people are more muscle insulin resistant and so on and so forth. You're going to have combinations of defects, of course. It doesn't have to just be one type. And then what we've done next is actually started mapping food responses to this. And it turns out, looking over here, same figure I showed you before, if you're insulin resistant, this is looking at the group as a whole, you tend to be a potato spiker, but not if you're sensitive. The same for pasta. And so we're seeing if this relates now to some of the other subphenotypes as well. So we're trying to match food with both your in this case, your mechanistic defect. And we want to do this for molecular markers as well. So we can type people in at the end and say, well, here's your type. You should watch out for potatoes. You can eat this and avoid that. The other area we've been pushing very hard on is this idea of remote monitoring. And so we've spent a lot of time on this, the idea of collecting samples. These are the Mitra devices. There's another set we've been using called Tassos. You put on shoulders, and then you overnight them to the lab, and then we'll do deep data measurements. And in our initial paper we published, we were measuring about 2,000 analytes. Now we measure about 7,000 with the later omic assay. So you can go really quite deep with this. And then different omics. And the proteomics and metabolomics are generally, the analytes are pretty stable on this matrix. For the lipidomics, it's a mix. Some are, some aren't. We know which ones are, we know which ones aren't. And we have targeted assays we run as well. And then we've run some really fun experiments. So this is where we had 32 people drink this shake that you can buy in CVS and Safeway and places like that around here. And then we profiled them. They were fasted before and after drinking the shake at various times. And what we discovered is that hundreds of molecules change, of course, after you drink this shake. But we can follow them, and we can follow some pretty important molecules like insulin C-peptide, insulin itself. This is one of those incretins. And we can follow inflammatory markers. And the cool result is shown here. This is now relative ion count, if you will. But in a general sense, everybody responded to this food differently. So each box is a different person. And this is just showing 3 carbs. Again, we're measuring hundreds of analytes. For this person, their carbs plummet after drinking the shake. This one goes up, 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 up. This one flat, this one down, this one over here down. And we can classify people into 5 groups of people informatically. And so this is the averaging, in gray, of all 32. And these are classes of compounds. So these are inflammatory compounds. So for this group of people, their inflammatory markers goes down after drinking the shake. Ugh, a little sensitive there. For this group of people, they go up. So same shake, it's pro-inflammatory on some, anti-inflammatory on others. And that makes sense because people have very different responses to food, and 10% of people have irritable bowel syndrome. In most cases, not clear what's causing it. We can actually map that out. And then this is where I want to have this field go, maybe not in quite this detail, but basically this is one person who gave a sample every hour for 7 straight days during waking hours, and while wearing a CGM, a smartwatch, and food logging, etc. And basically, the idea here is to correlate activities people do with both their physiology and their biochemistry. Okay, so what you do causes what? It's your biochemistry and physiology. And the net result is we could find thousands of correlations. We do something called timeline correlations. Find thousands of associations between people's activities reflected by heart rate, glucose, and steps with their cytokines, their lipids, their metabolites, and so on. And basically, there's a lot of fun stuff in these data. So we can see, for example, the insulin C peptide goes up after glucose, that you would expect, but we can measure it, which is nice. We discovered a new inflammatory control marker, transcription factor, that also goes up, we think might be responsible for the inflammation response of glucose. And then here's a fun one of alpha-synuclein, showing a very interesting pattern we think might correlate with stress. And some of you may know this is a marker for dementia and Parkinson's. And so perhaps this is a useful assay that could be used for potentially, having people maybe modulate their alpha-synuclein levels. It remains unclear whether a whole blood alpha-synuclein is reflective of what goes on for dementia. So that has to be worked out or looked at. Anyway, we can bring in all these data into a dashboard so you can see this wearable omics and clinical data. And then the ultimate goal, of course, is to integrate this into a hopefully health management system where we can basically bring in genome sequencing, omics profiling, or at least deep biochemical profiling wearables to better track people's health. Now, as I mentioned before, we do spin-off companies. This is our way of trying to get this out to the world. And what I want to tell you about is this one that does January AI is a CGM company. It's kind of cool. That's free. I have conflict in everything I'm telling you. I'm sure you appreciate that. January AI will help follow your glucose levels. This one's interesting. This is the microsampling, where you can actually mail in a microsample, collect a microsample, mail it in, and they'll give you a profile of 600 or so metabolites spanning these 20 wellness categories, things like inflammation, oxidative stress, and various, heart health, bone health. And one of the fun ones is the aging part, which I think is actionable information. You can get your aging, not only your biological age, so your biological age relative to your chronological age. So this person, for example, their biological age is 53, but their age in years is 50. So they're older than they should be. But the nice thing about these assays, which I think takes it beyond the methylation assays, is you can break it down. You should be able to set this up for proteomics, by the way, too. You can break this up. So this person's fine on their inflammation, not too bad on metabolic age, but their heart age is older. And the nice thing is there's actionable information behind this, and you can use AI to take data from the person as well as the data that's out there and make very, very specific recommendations, not just, exercise more, eat better, but they can tell you exactly what to eat, especially based on your existing diet, eat more of this, avoid that, so that you can ideally improve your markers. And 95% of people do improve their markers between the first and second visit. So we think it's quite powerful. I think because we're got a little bit behind, I'll wrap up. But bottom line is that we think deep data is going to be powerful. And I hope at some level we can integrate it into health monitoring and into our healthcare system. And a big factor that will, in fact, be, I hope this remote monitoring. And then I've been fortunate over the years to have an amazing group of people. And this is just some of the ones who have worked here. You're going to hear from Lee Watt and the single cell proteomics and her team later today. I have a close collaborator with Trace McLaughlin. This is the CGM group. This is a portion of the wearables group, the microsampling group, and this is the AI genomes group. So I saved a few minutes for questions. If people have some, I'd be happy to take them. So off we go. Otherwise, so.